Anytime you're ready.
Okay, well, thanks everyone for having me talk. This is actually really an exciting project. I say seeing is believing. This is slide number 1, the intro slide for those on the PDF file. Imaging HIV persistence. And this is really work from not just myself, but Henry Van Brocklin, who's our radiochemist, and Dennis Beckford, also in the radiochemistry department, really made have the busiest HIV-related PET imaging program in the world, I hope. Unless I know of other people that are doing this clandestinely, but at least from what we know, we're kind of really trying to set the agenda of what should be seen and how we can go about and apply some of these new technologies to advance curative studies, but also just our understanding of persistence and potentially even looking at new areas, therapeutic areas as well. So going on to the next screen, as you can see, we certainly have leukapheresis people, many participants, not just here at UCSF, but all over. We've also done lymph node biopsies. Sorry for the gross picture, on the upper right-hand side, and also gut biopsies as well. And this is really how we've been trying to get at where HIV persists, in what tissue compartments, to what level, what's the characteristics of those compartments, how much inflammation is there, et cetera. Something that we have essentially created, at least in people. Now, animals are different because you can obviously do more invasive studies in animals, but our understanding in people, who are definitely unique, is from very limited amounts of tissue from limited number of sites. So this has been a major issue over time. If you go to the next slide, these are just a couple of images from this initial paper back in 2015. It's almost 4 years now. Wow, it seems like yesterday. Looking at PET imaging in non-human primates. In this case, it is macaques, looking on the left side before antiretroviral therapy after an SIV-infected macaque, and then after, and seeing that they can see areas of what we call PET signal. This is the orange and the red and the yellows, these kind of heat map style colors, showing that those are likely areas of HIV persistence. Now, this was only in viremic monkeys. It was a very limited study, but it was really the first time that we would be able to get a snapshot, if not just a snapshot, but a 3D reconstruction of the total body burden of HIV in at least a viremic monkey. So this really kind of set the precedent, started a lot of excitement. I like to show the Where's Waldo slide. You guys have seen this many times, I'm sure, so I apologize. It's the same Where's Waldo slide. But if we try to cut out part of this this diagram or this picture to try to find Waldo, we're going to miss Waldo, but we're also going to miss that HIV particle or provirus, whatever it is, in the upper right-hand corner, which is very small to see in green. So clearly we need better things. I like to show the Where's Wookiee analogy as well, because if we're looking for Chewbacca here in the midst of the, I guess it's Tatooine, whatever the desert planet is in Star Wars, we're trying to find the Wookiee here, and it's very difficult to do so given the amount of background noise, the amount of other things that can really interfere with trying to identify that Wookiee. But as soon as we potentially do direct labeling of that Wookiee by PET, for example, then we can start to see them emerge over time. So that's really, really key. So I want to go over the overall PET goals. I apologize what slide numbers this up for the PDF folks. I'm sorry I don't have a number, but it says overall PET goals, and essentially this is what we're trying to do here through DARE, but also through AMFAR and through some other industry-related studies. But overall, this kind of unifying unifying research agenda that we're trying to do, essentially. So first is we want to perform PET imaging in HIV-infected individuals and uninfected control participants as well, both using direct and indirect markers of HIV persistence. I think this is really key. Direct markers are going to have troubles with sensitivity. There's not much HIV protein that's expressed in the setting of long-term antiretroviral therapy, so it can be very difficult using these types of analogies. But if we also have non-specific but more sensitive ways of looking at persistence, that's also definitely necessary. And I'll show you some data on both of those that we've generated so far in over 25 individuals. We're also correlating PET imaging findings with blood and tissue markers of HIV infection and immune activation. And I think our ultimate goal is really to do a combination imaging approach where we can look at these HIV-specific along with other measures that look, for example, T-cell activation, these more sensitive but less specific tracers that can really inform on what's happening in the reservoir on antiretroviral therapy. So ongoing studies, this is just to let everybody know what we're doing here at UCSF. We have a collaboration with Merck where we're using raltegravir. This is a radioactive raltegravir that Henry and Dennis have made, and we're giving it to those individuals both with early and later treated HIV infection. So those who started very early on ART, those late. We're also doing something called FARA-G. That's the second bullet under DARE. This is a directly DARE-funded project. This is a pilot study. This is a specific marker of T-cell activation that we are looking at and having some very, very interesting new results. I'll talk about some of the difficulties of running an imaging trial in just a few slides. And then also under AMFAR right now, but hopefully, you know, as we go further to find other support, the VRC01 imaging in vivo. And VRC01 is a monoclonal antibody that binds to the envelope or GP120 portion of the HIV envelope. So very much akin to what we saw with those non-human primates on the earlier slide. And again, non-human primates have traditionally been used, but now we want to go into humans. But not only do we go into humans, but we want to use the very latest technologies involved. And we'll talk about the Explorer project with UC Davis as well. It's really cool. I'm actually really excited about this. So a little bit about what we do. We label compounds, whether they're monoclonal antibodies to HIV proteins or whether they're drugs or small molecules, and we label them with an atom that is radioactive or atoms that are radioactive. Fluorine is one of the ones that we commonly use. If anybody's had a clinical PET scan, that's usually FDG, which is a fluorinated glucose. So it's a sugar molecule that has this radioactive fluorine. It has a very short half-life. We give this and with 4 hours it's gone. So we have to give and then image. So it's one of those things where it's very low radiation exposure over time, but we can give it and then we just image right away. But we've also moved on to something called zirconium. Everybody was a cubic zirconium is in fact fake diamond. It's very hard. It looks very much like real diamonds. It's cost effective. So I like to tell our participants that are actually getting some fake diamond into their bloodstream, although obviously we're not using real diamond. Couldn't afford that. But zirconium has some similar properties to carbon, but essentially it is a very long-acting radioactive isotope that we use, this 89 zirconium, which has a half-life of over 3 days. So it actually sticks in the system for about a week, and that allows us to do serial imaging over time. So we can look at dynamic changes to an intervention, for example, to therapies. And we also allow various drugs, or in this case, monoclonal antibodies, to permeate those tissues, to really spread and to get where they need to go. I think that we're going to show data that you may need 3 to 4 days before you're going to start to see a signal in any of these type of modalities. And that's going to be really important. Okay, so the next slide, this is called tracer dosage and imaging. Now, I think I want to talk a little bit about the safety up front, because we are giving radioactive material to people. And to people that are otherwise healthy. Some of them are uninfected. Many people are on suppressive ART. We also have viremic individuals. But either way, we are introducing potential risk. However, the amount of drug we actually give, now this is not the radioactive amount, but the amount of drug we actually give is tiny. It's almost homeopathic. It's like going to the store and looking at some of those kind of homeopathic remedies. We're giving a tiny, tiny, tiny dose. For example, this FARA-G molecule, which binds to activated T cells, I'll talk to in a second, we actually give it at 1/1,000,000th to 1/100,000th of a clinical dose. So they really have no overt biological activity. So the compounds themselves, despite what they're derived from, are usually incredibly safe to give. So they have negligible biological activity, and they're actually very well tolerated, as we have already discovered from our initial studies. I should also mention that we are trying to be very inclusive with enrollment, especially trying to increase women in our study. We have imaged and enrolled women already, although it's certainly lagging behind the men in our study as well. It has to do somewhat with the population here in San Francisco, but we're certainly trying to reach out and to be inclusive with our enrollment. And the other thing I'd like to say is that no one knows if these are clinical trials or not. So we've actually been in discussion with the NIH, but also with other groups about, whether giving someone a radioactive tracer is actually an intervention, because we're not studying the effect of the tracer. The tracer has negligible to no biological activity. We're giving a compound just to see something. It's like doing when we do a lymph node biopsy, we take a lymph node and then we look at it, so we're not actually changing the biology of the person. We're not looking for, like, for example, the antiviral effect. We don't have nearly enough drug to do that. But at the same time, there's potential risk from radiation, and there is potential risk from radiation. So, but then the FDA is more concerned about risk than they are about whether this is a clinical trial, so they really don't have any say in that. And I think what we've all been thinking, and I think that where the field is now evolving towards, is that in fact these are not kind of interventional clinical trials. These are they're clinical studies, but they're not clinical trials themselves. We're not looking for an outcome here. We're just giving an agent and imaging someone to see what it looks like. However, we had actually been put on hold for a while for one of the DARE projects because we have to undergo the CRC review at DAIDS just to make sure that we are doing this correctly. We didn't actually knew that we needed to do this earlier because it was not considered, or we did not consider this a clinical trial. And I think people agree with that, but they'd still like to have it reviewed. So we're just on hold right now until they review. I don't expect any difficulties. It's just a matter of having them kind of officially approve the process. But we've already started the study and actually have quite a bit of experience thus date. So the next slide, this is our workhorse here at UCSF. Most studies doing imaging. This is our PET MRI. This is a combined MRI machine, which is does not include any type of radiation. It's an MRI, so it's a very strong magnet. So people with a pacemaker or metal, it might be difficult to image. But essentially gives us very detailed views of the tissues within the body, the anatomical structure. We can see water versus bone versus muscle. We can look at different tissues. We can identify lymph nodes. We can look at the brain. We can do other things with the MRI alone. And then there's also a PET detector within this machine that looks for radiation exposure. The only radiation in this study is given in the tracer through the vein. It is not from the imaging modality itself. These are purely non-invasive imaging modalities. Although whoever's been in an MRI machine before knows it can be very claustrophobic. It's loud, it's noisy, and just some people, including myself, just can't tolerate them. So it can be a challenge to do that. But they're not invasive, and they don't give any radiation. It's just a matter of a strong magnet for the MRI machine. The next slide, these are some other things. We also have SPECT. These are different modalities of looking at radiation. We have the PET CT, which is kind of like an MRI, but it's a CT scan, which is much quicker. The CT part is. It's a much smaller tube, but it also involves additional radiation. So we've been avoiding the CT because we want to keep as low radiation exposure as possible for these studies. So it's very, very, very exciting. If you go to the next slide, I want to show you the first few slides. And now some of this data has been seen before, but I'm going to curate it here. And this is our very first imaging study that we've done, and actually probably the last one we'll finish. But this is the labeled raltegravir. We were interested in knowing where does raltegravir distribute when it's first given, especially through the IV. So it's bypassing the oral metabolism. When we give raltegravir parenterally, where does it go? What is the distribution of the drug? Is it getting to the tissues that we need it to get to? Is part of the reason that we're seeing rebound off of antiretroviral therapy is because antiretroviral therapy isn't getting to certain locations, and we're just not able to sample those locations. Hence, we don't know it's not getting there. For example, the brain or from other areas. So, so far, we've imaged 3 participants. Our goal was to complete imaging in 5 chronically infected individuals that started therapy late in disease or late in infection, and 5 who those were treated very early, within 4 months of infection. And the thought with that is that it's possible that the lymph nodes may fibrose over time if there's a delay in ART initiation that may actually reduce absorption of the drug into those tissues, and that could be a bad thing overall. So this is our first study. I'd like to tell a quick story about our very first participant who we imaged. And we were very excited. We were very anxious and nervous because we'd never done this type of study before. We have in cancer, but not with our favorite population here at UCSF. He was all set. He was in his gown. He had his IV, and he was about to go into the MRI room, the PET MR room, and get his injection of raltegravir. And he turned around and he looked at us and he said, "Light me up." And then he turned back and walked into the room, and we were all very happy. I mean, there was just so much interest and excitement, not just from us. I mean, honestly, there were like 5 of us standing around from the entire study PIs, and we were just like, waiting for the images to come up in real time. So I'm sure that didn't make him feel any more less anxious about participating, but he was really fabulous. And I think this has been the overall reception so far. We've had no trouble finding at least ART suppressed individuals or uninfected controls. We've had a huge amount of interest. I think the challenge has been finding folks that have high viral loads. For understandable reasons, but we've actually had a very easy time. Our radiology collaborators are shocked. Like, how do you enroll an imaging study so quickly? I mean, it takes them years to enroll a study. We could enroll a study in 2 weeks. With our Scope, with Becky and Viva and Enrique at Scope, who've really done an amazing job kind of reaching out to people and educating people and getting folks interested in our study. And also, we've had a lot of unsolicited interest as well. So it's been really fun. I mean, we're really having a great time with this study, and I think it's really going to help. So anyways, let's go to the next slide. This is what the images look like. Now, if you're not a radiologist, which I am also not, you may wonder what I'm showing. So I'll show you a schematic in a second. On the right, that is someone's head. And what we're doing is these are cuts. These are called axial cuts. It's like we're doing slices, horizontal slices to the ground, if that makes any sense. So for example, here we would be slicing through the ear to the nose. You can see the nose on the top there. It's like someone's laying down with their head facing up and their nose sticking straight up in the air. That's their nose up there. You can see some of their eye sockets, and then you see that big gray area, which is the brain. And what I think is important to see there is that we see these little red areas, and the red areas is the uptake of the tracer. That's the PET tracer. That's the raltegravir coursing through the body. Those are blood vessels. So we know that raltegravir is going to be in the blood because we gave it IV to the blood. But what we're not seeing is we're not seeing any red in the brain tissue itself. We're just not seeing it. Now, if you look on the left side of the slide, this is now cutting through the pelvis. So this is not through the head, but it's, again, a horizontal slice to the floor, and it's going through the pelvis. So if you go down and you feel kind of where you feel your pelvis and you kind of work your way down to the hip bone, that those two little dark things that look like eyes or butterfly wings, those are the balls of the hip joint. So this is kind of cutting through the lower pelvis. So you can see the hips. And what I'm pointing with that white arrow is actually a lymph node. I don't know if that's an inguinal lymph node. So that would be in the inguinal area, in the groin area. And what we're seeing there is a lymph node, and that lymph node is cold. It is not seeming to have drug delivered to it. We've looked at this now in several individuals, going to the next slide, and we've also seen how much radiation each of these kind of systems get. It turns out that the things that are getting drug are the small intestine, the gallbladder, but that's because we know it's metabolized through that system as well and excreted. But we're seeing very little in the brain. We're seeing little in the spleen, little in the thymus. So it kind of, organs that we think are important for persistence outside the gut, of course. We're just not seeing a lot of raltegravir uptake. Now, this could be just because of the tracer dose. It's a very small dose that we're giving, but it glows pretty brightly. It could be the timing. We have to image within a few hours of injection. If we waited longer, maybe we would see better penetration, but we just don't have that technology with the fluorine molecule itself. But I think this is very informative, at least from the negative data so far. Now, I should say this is a preliminary study. We haven't finished. We have 7 more to go. So I don't want to make any broad generalizations here, but basically this is the type of information that we've gotten from the raltegravir. So the next slide, we can kind of skip over this, but this basically tells us that in order to get raltegravir into the brain, you need to have inflammation of the brain, and so far we've only imaged folks that have been on stable ARTs and should not have inflammation. So it's not surprising that we're not seeing a lot of drug this early. Okay, next slide. So now this is where it's getting really fun. And very kind of exciting for all of us, is this VRC01. This is the anti-GP120 HIV envelope monoclonal antibody from the VRC. The NIH has graciously given us this molecule to use. And what we've done in this complicated, everybody remembers their organic chemistry, which I do not. But what we've done is we basically tagged this large molecule onto the monoclonal antibody, which is the Y, and onto that we've put this zirconium molecule. So each antibody gets a single zirconium molecule that is radioactive for about 5 to 7 days. If you go to the next slide, these are not people; these are mice. This is what a rodent looks like in a PET CT. This is a small rodent PET CT, but this is basically our very first biological administration of this labeled VRC01. And we just do this into mice and rodents because we need to know for the FDA to get approved to do it to people how much radiation is going to be exposed. And we actually use this rodent model to look over time, and you can see at 1 hour, 24 hours, 48 hours, and even 120 hours. So even days after infusion, we're still seeing some of this radioactive uptake in the mice. And this is, from this we're able to calculate how much radiation to each organ, and that way we know that we're giving a safe dose when we first give it to patients. So this is something that we would not give to humans without doing these types of studies first. It's really for safety. I don't work with a lot of animal studies. I prefer to work directly with people, but these are certainly needed just to make sure that we're doing the safe thing when we move into the participant. Okay, now, next slide. Well, we also have this excellent technology called Explorer, which is completely nondescript, but sounds cool. This is what we call a total body ultra-high sensitivity PET imaging. There are only 2 of these right now in the world, and one of them happens to sit about an hour up the road at UC Davis. Depending on traffic, could be an hour and a half or 2 hours, could be 45 minutes, depending. But this is pretty close by, and what this is essentially a super PET. So we are able, not only, it's like having almost like, I don't know, like 100 PET machines all in a line, or like 50 PET machines scanning all together at once. So it's like this, it's really sensitive. It can actually increase sensitivity by about 40 times. It can increase the signal to noise, which is, the signal we care about versus all that vascular noise that you saw earlier, all the red, by about 6 times, which is kind of dramatic for PET scanning. Obviously, it has higher fidelity images. It's kind of like comparing an old monograph record from the 1920s to, a modern kind of Blu-ray, disc or something like that. So it's really amazing what kind of advancements that have been made. We can actually use lower doses because it's so sensitive. We can use less radiation, which is awesome. So we can actually give less radiation to people. We can actually track the fate of radio tracers for a longer time after injection because it's more sensitive. So we can look at that decay rate longer, and in fact can scan within about a minute. So although you have to go all the way in it, it can actually scan in about 1 to 2 minutes, and it's done. I mean, it's really fantastic. It does not have an MRI machine attached to it. So that is one of the downsides. But if you go to the next slide, what you're seeing here are different cuts. This is from the left. This is like a side slice. If you slice someone in half when they're turned to the side to you. This is a front slice of a person. Then these axial or horizontal slices again at different parts of the body. And what you're seeing here, this is FDG. This is not our compound. This is just from the Explorer program at Davis. But seeing that this is a glucose molecule that's been labeled with that fluorine, and it almost looks like a CT scan. There is so much. We can see individual tissues and structures simply by PET scan, which has never been able to be done before. It's always been coupled with an MRI or a CT scan. But here we have that level of clarity with just the PET tracer itself. So it's really unbelievable. And we're actually looking forward to using this Explorer technology. We've actually already used the Explorer technology, not in people, but in non-human primates up at Davis. These were uninfected macaques that we gave this VRC01, again, to try to look at the safety of the radiation exposure over time before we gave it to people. And for those looking at my screen, I will animate it for you. I'm sorry for those with the PDF, but you can see these kind of monkeys spinning around. And this is day 0 and then 6 days, actually, a long time after administration. And you can see that big blob in the middle. That is the liver. Oh, I just lost one on that one. Okay, whoop, I lost the other one. Anyways, we'll go back there. That's the liver, and that's where it's metabolized, but you can see it coursing through the blood vessels, but it's starting to accumulate in various tissues as well. So this is kind of what we can expect when we move forward with people to really try to localize. Now, these are uninfected monkeys, but we just wanted to know what the radiation exposure and if we could actually use this technology. Okay, so moving on to the next slide. As you can see here for the blood clearance, I'm just showing a graph that, we have this fairly stable period of tracer for about 5 days, starting 24 hours after injection, where we can start to image. So we really have a long period of time. We can give some a tracer and 3 days later send them to Davis for Explorer imaging. So it's really kind of a powerful tool. For example, we could give tracer, wait a day, give a reservoir reactivating agent, and then scan in 2 days, and we could probably see a change. So that's kind of what we're planning going forward. Here's the next slide. Here's just showing, again, these our very first human VRC01 images. And I want you to appreciate on the left is an HIV uninfected controlled participant. The person on the right has a viral load, but not very high. It's about 20,000 copies. So kind of a viremic controller, semi-controller on the right. And you can see that at least in the gut, there seems to be a lot of initial uptake. Some of this is actually just excretion from the bile. but some of it is actually a collection in the intestines as well. But you can also see there's a lot more noise overall. These are just some very overview pictures. I'll show you some. This is just the PET itself without the MRI, but I can show you both together next. But you can start to observe a few differences. And it also looks darker in the vessels as well in our individual that has some low-level or some mid-level viremia. Okay, so the next slide, this is what we're going to be looking at from now on. So those are easy because you can tell what they look like, kind of look like a person. But what we're going to be focusing on is on this green slice, this transverse plane or this axial imaging. And you can see that in this kind of animated participant, that if you kind of slice them where that green plane is, it's parallel to the floor, horizontal to the floor, you're going to get something that looks like what you see on the right. Kind of like you're looking up into the body from the floor. Is kind of the analogy that we have. And there we can see on the bottom is the backbone. We can see some of the ribs. Those aren't ribs. I'm sorry, just yeah, there are some ribs there. There's the liver. There's kidneys, are those two things on the bottom. So anyways, it gives us an idea. Don't worry about the anatomy all here. It's the PET imaging that matters. Okay, so here is our kind of results from our first in human PET VRC01. This is our HIV-specific monoclonal antibody. On the left is a negative control. You see that there's still some red and some of that red-white heat that we're seeing, and that's the background, that's the noise, that's just the tracer going to where it's going by itself without being directed to HIV. But what you can see here is that I'm circling right now is over again, it's hard to see, but this is a lumbar vertebrae. This is one of the columns in your backbone that I've circled. And what you can see on the right is the intensity of the signal compared to the intensity signal on the left. And this is 3 to 4 days after we I think it's actually 3 days, I'm sorry, not 4 days, but 3 days after we gave the tracer. And you can see that 3 days later, we're still seeing a huge amount of signal in the bone marrow, albeit a kind of a mid-level viremic participant. But we certainly see a difference in signal. We're also seeing more uptake spread throughout the gut as well, where we would expect to see HIV. And so we're hoping that what we're seeing on the right is an HIV-specific signal binding to HIV protein. So that was really, really interesting to see that we were able to see this difference up front. So if you go to the next slide, okay, so here's a little bit farther down in the pelvis again in one of our male participants. I have a little box. Underneath, I have something called the negative control, and it looks like they're also lighting up red. But if you look at the box, there's two structures there. There's the thing on the top, which is a lymph node. Underneath the lymph node, there's a very bright collection, and that's a blood vessel. Those are the arteries and veins that go from your pelvis to your leg, the femoral artery and the femoral vein. Those are lighting up in both the negative control on the bottom, as well as the HIV-infected individual. This is, again, someone with about 20,000 copies. But what I zoomed up on the upper left hand of that box on the right, you can see that at day 1, so this is after they first got their tracer, there's a lot of signal in the blood vessels, but there's not a lot of signal in that large lymph node, a few kind of red dots, but that's noise, that we see on the upper left. But if we go 4 days later and we look, now we see that lymph node lighting up. It's almost like a light bulb. Like we see this large signal, but it's interesting. It's also not a homogeneous signal. There's this kind of heterogeneous but very strong signal. At the same time, we're seeing a decrease in the intravascular signal from this participant. So we're really seeing a change over time. We're seeing an increase in the lymph node of this viremic participant, but a decrease in other areas, suggesting that the tracer is going to an HIV-infected area that's expressing protein. This made us very excited that we were able to see this. I should mention that the first day of imaging is about it's about an 8 to 9 hour day. It's maybe like 9 or 10 hour day. It's a very long day. We're now done with that. Going forward, it'll just be an hour day each. So hopefully, it'll be much easier. But these initial participants were troopers. Let me tell you, they sat waiting for 3 different scans over a single day. I actually went out to the Safeway, which is our local supermarket, and bought lunch for everybody because people were starving, and they have IVs in and they're radioactive, and we didn't want them to leave the room, and so it turns out that most people have ordered the same exact meal. So there seems to be a shared love of the Safeway spicy wings amongst our population. So anyways, it's just a small thing that we could do to help people more comfortable, but it was really fun to kind of be there with them and kind of talk about their experiences as they're undergoing the imaging studies, because we had no idea what we were going to see on the scanner either. I should also say that there were several of us kind of leaning over the radiology tech who was doing the imaging at the computer, seeing the images as they came up in real time, and he probably thought we were all crazy. Because we were just sitting there staring at it with so much intent. But I think everybody's been having a lot of fun with this, although it's been hard to get off and running. Okay, the next slide. So these are our initial 4 individuals that we have actually done 8 so far, 4 uninfected and 4 viremic participants. We wanted to make sure we were able to see something before we started imaging the larger population. And we have people with different viral loads, over 200,000, around 100,000, actually it's around 70,000, 20,000, 2,000. And then uninfected controls. So we have a range of different viral loads. And what I'm just showing you here are the initial 4 that we've been able to analyze so far. And we see that, like, for example, in the bowel on day 1, we see very little uptake on our controls, but we see a lot of uptake in our viremic participants. If we look at bone marrow, again, we're seeing a much higher signal in both our viremic individuals with the bone marrow, and at least in one of them, we're seeing a lymph node signal. In fact, in the one in purple, the lower level viremic actually didn't have any noticeable nodes on MRI, so there was nothing really to look at. But there were some nodes on some of the control individuals, but they did not light up. So that was good to know. So anyways, we're starting to see signal. Again, next slide is going to shift focus, for example, but we've actually imaged 8 individuals so far. AMFAR has been a little bit stickler. They wanted us to do 5 viremics because that's what I initially said in the grant, although I just made that up number. I mean, I completely made it up. I don't know where it came from. And I think we have enough information now that we can actually continue on with ART suppressed individuals, because that's really the target population of interest. Now that we know that we can see and visualize virus using this PET, we want to do it in aviremic suppressed individuals, and also using the Explorer program at Davis. But we just need to convince our funders that it's worth going on to. I think we have plenty of data to suggest that, but we'll see what they say. So we're going to put that together in the next week or so and try to make our case to move forward. So anyways, we're not just doing, that's an AMFAR funded project, but this is the DARE funded project here. And what we're doing is we're not just looking directly at HIV in the body, but we are looking for other markers. So for example, we have this molecule called F-Arag. And I can tell you it's the full name. I won't tell you because I would have to look it up each time. It is also known as Nelarabine, which is a chemotherapy agent. So it's actually a cytotoxic chemotherapy agent, which gives everybody pause until they know that we're using 1/1,000,000th of a treatment dose, which is very low. It doesn't have any chemo effect, in other words. But what it does is it's selectively uptaken, and this is a completely uninterpretable cartoon, so I apologize. But what it's done, this is a T cell, it's taken up by activated T cells, and it's trapped in T cells that are proliferating or activated. So essentially what Nelarabine is used for is T cell lymphomas. T cell lymphomas are T cells that have gone haywire and are proliferating and are very activated, and that's why they use this compound. It selectively collects in those T cells and kills them. Now, we're using a very small dose, so it selectively accumulates in the T cells but doesn't harm the T cell, and we've tagged this with the fluorine. So again, it's a shorter acting. We can't do the 3 to 4 days that we were doing above, but we can at least look where at the time of injection there are activated T cells in the body. Now, we already know that there are activated T cells in areas where HIV persists. So that's something that we already know. Peter Hunt and many others have shown that. We also know that individuals with HIV, even despite antiretroviral therapy, have foci of elevated T cell activation. They have lymphocyte activation, and we don't exactly know why that's the case, despite the fact that we're giving suppressive antiretroviral therapy. So we thought it would be really cool if we could image T cells, not just through gut biopsies or lymph node biopsies, which survey a tiny fraction of the lymphoid cells in the body, but we could do a full body whole survey all at once within an hour to see where activated T cells are. So we thought this would be really cool. It's actually being developed for cancer. And the next slide I'm going to show you, I just put up that's a mouse that had activated versus non-activated T cells injected into it, and then the imaging showing the activated T cells glow brighter. That's pretty much just the proof of concept. But if we look at the next slide, now here on the left, our pre-HIV population imaging. So now we're seeing a healthy control on the left. Now you see the liver, that big blob on the left, is the liver. The big blob on the right is the spleen. And then you can see the backbone there, those little segments, and you can see the brain is pretty clear and white, doesn't seem to have much signal in the brain in a healthy individual. And now what we have on the right is a participant with metastatic breast cancer after getting anti-PD-1 therapy. So they got immunotherapy, which allows activated CD8 T cells to go to areas of tumor and attack them. So what we've done is image these breast cancer patients after they've gotten the anti-PD-1 or CTLA-4 therapy, or as a combination, I can't remember, but either way, those T cells are now homing in on areas of tumor. And what you can see is in the bones, especially in the backbone down there on the lower part, in the skull, things like that. Those are where your CD8 T cells are going. Those are activated T cells, and they're going to areas of tumor. On the right, I show now a PET MRI, not just the PET, but the PET in red and the MRI for anatomical localization. And this is, again, one of these axial or kind of horizontal floor cuts through the belly. And what they're seeing here is actually that's bowel. That's a loop of bowel that's lighting up, suggesting there's a metastatic foci in the bowel as well, or at least there's activated T cells going there, which is kind of an indirect measure. So given the success of this agent for tumor imaging, we thought that we would definitely want to try this in our HIV population. And that's why we designed this protocol through DARE, because that was one of our main, that was for our IRF2. One of our main projects was to image even indirect imaging of reservoirs, and this is exactly what we're doing for IRF2. And what you can see here, again, these are these axial images, but of the brain. This is the head again. The nose is on the top. Those kind of spaces underneath the nose, where the nose should be, those are the sinuses. So those are the big empty air spaces in your head that get filled up with phlegm when you have a cold. There's a lot of space in there. That's why you can have a lot of snot. So anyways, what you can see compared to the HIV negative participant on the left, that's B. We kind of took this from a grant, so it's not AB, it's like BC, so I apologize. But and then you look at number C, and you can see that it's a little subtle, but there is background uptake. So we know that there's this background noise that we can see with the negative control participants, why we have to image these folks in parallel. But on the right, I don't know if you can see this, but there's these areas up on the top there, but also down in front of the brain, in front of that cerebellum of the brain, and those are glowing much brighter. And that's actually the nasal associated lymphoid tissue, which is a known reservoir for HIV, and we're seeing actually an increase in T cell activation. Now, this person had been on ART for at least 10 to 15 years. Was infected in I think the late 80s, early 90s, but had been on long-term ART. And even then, after years and years, over a decade of therapy, we're seeing persistent residual T cell activation in areas where we know HIV persists. So whether these are related or not, obviously, it's a correlation, it's not a cause, but I think it's definitely worth moving forward and investigating. If you look at the next image, this is a little bit more difficult to understand what I'm showing, but this is, again, a frontal image, kind of looking front, like you're staring at the person. That big, all the black stuff in the belly, that's the liver. And the spleen, and then that's the bladder way down. That little ball at the bottom is the bladder. So they're actually peeing out the tracer, and we can see the tracer in the bladder. But what I want to show you, this is an infected individual, again, ART suppressed, and the arrow is showing that you can kind of see the backbone. So you can actually see these different vertebrae that are going down the spine. Now, we shouldn't see that. We don't see that, but we see a little bit of it in our uninfected controls. But what we saw as well is that the bone marrow in our infected individuals, whether they were viremic or not viremic, actually had an increase in T cell activation that we imaged by PET. And it was exactly the same thing that we saw with the anti-GP120 antibody. So we're seeing HIV persistence in the bone marrow, at least what we think is persistence, and we see what we at least think is T cell activation, again, in areas where we know HIV may be persisting, although no one's really looked at the bone marrow. It's hard to get. I think it's uncomfortable to do a bone marrow biopsy, but I think we've been overlooking the bone marrow. We have activation. We have T cells in the bone marrow. It should be B cells, but we're seeing T cells. They traffic through. They're being there. Are they there because there's HIV there? That's something that we need to ask. It's something that not many people have been pursuing. So anyways, another just very early days, just a few months of imaging, but we've already found new things that were unexpected. I want to show you again. This is another image showing that we even saw differences within ART-treated individuals. On the left is an early treated person. The circle is around one of those lumbar vertebrae, so that's bone marrow that we're seeing in the middle, and that's glowing nice and, well, it's not nice and bright, but it's glowing. And then on the right, we see a later treated individual, and we actually also see that glowing, but it's a little bit less. So we're seeing differences in the signal intensity depending on when individuals started intraviral therapy. We don't know what this means yet. We've actually imaged 9 individuals so far, all HIV infected. We've already imaged controls with our cancer cohort before. We're trying to age match those as best we can. Anyways, we certainly know that we're seeing kind of a similarity in this uptake. Again, one more graph. I apologize. Graphs are never fun, but on the top, this is our FARG. This is our T cell activation agent, and we're seeing all those colored dots are different individuals. For example, early ART in green, late ART treated in blue. We have viremic less than 2,000, like low-level viremic controller in brown, and we have this higher but mid-level controller viremic in red. And you can see that in our FARG, we're definitely seeing differences between the nasal-associated lymphoid tissue, the bowel, the spleen, which we hope we would see, the bone marrow, between our uninfected controls, which are all in the black circles and much lower. Again, I showed you on the bottom, that's the VRCO. These are not the same individuals, by the way. These are different patients because we haven't given them together yet. But anyways, this is to show that we're seeing similar findings and similar tissues between 2 very different modalities, but looking at a similar concept. So I think we're really excited about this initial data and really hope that someone will pay us to do more as we go. We're trying, we're writing grants and things like that. So I think one was reviewed today. I'm too scared to look at the score today. Maybe tomorrow I'll have the guts to look. But anyways, we also have applied PET imaging. If you look at the next slide, well, we're not going to stop here. Our job is not just to say, oh, look, we've seen residual HIV in the bone marrow or the lymph node, because, okay, that doesn't help us a huge amount. It does help us to know where it is, and we're going to get that sense. But we want to know how this T cell activation, this residual inflammation, correlates with residual disease. So what we're doing as we're going forward is we're using dual imaging. We're going to give FARG, image somebody, and then the same day, once they've been imaged, we'll give them the zirconium. We'll make sure that they're under the radiation limits, of course. We're being careful to do that. And then we can image them over time, and we can actually correlate directly at the same individual in the same week. In fact, in the tracer is given the same day. We're just imaging at different times. We're able to see like how these directly correlate. So that's going to be really cool. We're also looking at imaging of PD-1 expression, not just T cell activation, but PD-1 expression, and that's in the works as well through DARE, although we're kind of more in the earlier stages on that project. If you go to the next slide, we've convinced Steve Deeks that this is a cool thing, and that we have latched on to several AMFAR-funded clinical studies. We can't do a clinical trial with the money, but we can certainly latch on to existing clinical trials and image in that setting. And so, for example, this is the JAWS study that some people may be aware of that's giving, oh gosh, everything. It's giving an HIV DNA vaccine with electroporation, which apparently hurts. They're getting MVA boost, they're getting BNABs, and they're getting a TLR9 agonist. I mean, they're getting everything, and then they're taking them off therapy. This is, I really hope people will enroll in this study. It looks very intensive, but I think it's going to be an important study. Anyways, but there's also going to be a treatment interruption. The people will come off therapy and they will rebound over time. So we're planning on imaging at different stages in different individuals and even serially over time within our radiation guidelines. Again, making sure we're under those kind of FDA limits that were there. And we'll try to see what happens before and after TLR administration. If there's viral reactivation, can we see it using PET imaging? We're also going to be imaging during rebound, one of the AMFAR rebound studies, to see when people stop ART. We'll try to image everybody around week 2. Or 1.5, when about half of individuals are starting to come up pretty rapidly. And as soon as they are detectable, they are restarted on ART, and that actually helps us because it kind of freezes the reservoir before it starts to expand further. And that way we can image and get an idea of where virus was rebounding, not just where it persists, but what foci, what anatomical areas actually caused rebound, and maybe we need to target those better when we see how those are. So that's also in the works. I got to say this is more about 6 to 10 months away would be my guess, but we're still in the planning phase and we're actually very serious about moving forward. We're already getting an FDA document together to get FDA approval, and again, just looking at how we're going to fund this going forward is always the biggest challenge. And with that, I would like to just kind of stop and have time for questions. But I just want to thank all of the hipsters. This is our hipster study, not just because we're in San Francisco, but because it's the HIV, oh gosh, Becky Ho labeled it, HIV imaging and persistence, something by PET-MR or something like that. Anyways, it's like imaging HIV. So anyway, these are our hipster study. And on the left is our radio pharmacy and our radiologists who are helping us out, and then on the right, my lab as well, actually as of about 4 months ago, who's been doing a lot of the reservoir work in combination, and we're testing T cell activation, reservoir levels in the various tissues that we're imaging. So it's kind of a combined approach between this, and we've really enjoyed working together. It's the first time I've ever worked with radiochemists, and I love it. This is the first time I've worked with HIV docs, and I think they love it. I hope so. We're all a little bit crazy, as Steve Deeks would say, but in a good way. And I think we've all really kind of enjoyed learning from them. They've learned from us. We've also learned from the community. We've talked to individuals about who participated in the study, asked them what their thoughts were, what they're thinking, why they're doing this. We haven't been collecting it formally, but it's just been very interesting to kind of interact. I actually obtain consent from every individual when possible. I try to be there for the first imaging session for every participant, so I can really kind of stay involved and kind of learn from people and what their experiences are in this project. Overall, so far we've had overwhelming positive feedback. I mean, some people I thought was a long day and the scanner wasn't good and wasn't fun, but you know, we're trying to make it better for people as we move forward. And moving forward, hopefully we won't do several images over a 9-hour day, but we'll have 1-hour days, maybe twice ever, and that will be it. So like 2-3 hour commitment total, and hopefully that's where we're moving now. So with that, I will stop and see if there are any questions, comments. I would welcome feedback as well as to potential participants and what feelings are about being in such a study and what we could do to make this better or easier.
That's amazing, Tim. So you answered my safety questions, and you answered my question about how long it used to take and how long it takes now.
Yeah.
You said that it takes 3 days before you can see it, but then you said that you have to image within a few hours of the injection. So
yeah, I'm sorry. It depends on the tracer. Yeah. Okay. Yeah, the FRG we have to do right. So the T cell activation tracer, the FRG, we have to do right away. It's just the way that we've labeled it. We can't. It's a small molecule, so it's a short acting. But the HIV specific monoclonal antibody, that we can image up to almost probably 5 to 7 days. We're going to try going a little bit longer next to see what we see. But yeah. So they're different depending on the compound.
So I guess we don't know how much that machine costs at UC Davis, but I mean, what are the chances of getting a couple of them at UCSF?
Well, so the machine would be beastly expensive, but they have already committed to working with us. They are part of a grant that we submitted as well, and that we hope it's a possibility that the machine might reside at UCSF simply for ease of enrolling participants, which would be fantastic. If not, because the tracer that we're giving that we care about for the Explorer, which is the monoclonal, has we can give over days. This is something where we can give an injection, say on a Friday, and then we can arrange transportation to have someone take a car service to Davis on the Monday or Tuesday, spend the afternoon there, and then come back. So we're trying to make it as easy as possible. But either way, it's definitely worth the $60 trip to Davis and back for sure, because we think that this Explorer, this Super PET, is really going to revolutionize the way. And we're really one of the few groups worldwide that will have access to this. And I think that makes us very unique in the types of imaging that we can do.
And so do you think this is going to work for patients that are treated later? I mean, that was kind of scary.
Yeah, whether we're going to see something or not, I don't know. I think with some of the monkey experience that's been not published yet, but been talked about at meetings, it looks like the signal really disappears when you use monoclonals, when you start folks with long-term suppressive ART. However, it's possible that the Explorer would be able to provide signal. I think, when you have a 40 times increase in sensitivity, that may push it over the threshold to see something. I mean, I was shocked by the quality of those images. The other thing that we're doing too, Linda, is that we're trying to design studies that involve, for example, treatment interruption or other things that are not for the imaging, but that are being done for other reasons, and to latching onto those and saying, okay, if we can't see it during therapy, what we can do is we can image someone before they start, and then we can image someone just after they've come off, and we can see how the distribution of virus before they started as they were decaying versus where they rebound. And that will give us a clue as to where things are hiding long term, even if we can't see it directly. So we're contingency planning for that particular scenario. We're really trying to see when we can image individuals at that sense. Does that make sense? So
for the JAWS study, since it does everything but really measure latency, so
yeah.
And
Yeah,
my
they're getting tissue biopsies and all that stuff, so we can correlate what we're seeing with all the tissue levels as well.
right, last
Yeah.
question, this hold, do we know how long that's going to be or how long it's going to take?
For the FRG, what it should. We just have to wait for CRC review. I don't think SRC. I don't think it's gonna. I don't think there'll be any problems. I think that this will be supported. And there's no safety concern. It's not going to be an issue. But we had to wait for the company to sign a CDA with the NIH because it's an open review meeting, and the company wanted to kind of protect the information that's in their patent, if that makes sense. So, but that's now in place. So Henry and I are going to be submitting to the to DAIDS probably tomorrow. And whenever the next meeting is, we're hoping that we're going to get approval after that. So it might be a couple weeks from now, but we're hoping we're going to be up and running again very short. Again, there was confusion about whether this was a clinical trial and whether we actually needed review. We didn't think we did because it wasn't a clinical trial, at least it wasn't defined as the agency defined it. Although there are people in the agency who did define it as a clinical, it was just very confusing. I think what it told us is that we're the first ones to do this. So we knew we were on the cutting edge of science because we were challenging an established infrastructure that had no idea what to do with it. So that made me feel, it was frustrating, but it also made me feel like we're onto something good. So,
How about
if that
anybody
makes
else
sense.
questions?
Tim, from a participant point of view, I'm just curious, in your early slides, you had pictures of the device, and I'm not clear if it's a tube or if it's a pass through a hole.
Oh, I got. Is it Michael? Is this?
Yeah.
This? Yeah. Yeah. So it is. It is a tube. It's an MRI tube.
Okay.
It looks deceivingly like a hole
Yeah.
on the image. It is an MRI tube, so it does involve going in. Although it's not as long as some of the older MRI machines. It's about I would say it's about 4 feet long.
How long are they in there?
They're in for about 45 minutes, so it's not a short period of time.
Right. So as a person who's participated in trials and whatever for those type of things, I have severe claustrophobia. I have to do Ativan.
Yeah,
Do you provide that on request, or do you?
So we're personally not providing it, but we are allowing their primary care providers to provide it, or one of their clinicians. And what they'll do is if they can talk to them, say, hey, I'm going to be in this imaging study, can I get some Ativan? And several people have done that so far, and we're absolutely okay with that.
Right.
I need Ativan to go to an MRI tube as well, so I feel it.
That's a lot better than 8 hours, though.
Well, so it's actually only 45 minutes at a time. The 8 hour was the day. So what happens is they came in, they have vital signs, they have blood pressure. We just do a quick exam to make sure everybody looks like okay. We give the tracer, we put an IV, we give it, and then we wait for 2 hours, and then they go in the scan for 40 minutes. They come out, they wait 2 hours, they go in the scan for 45 minutes. So it's the scanner time itself is only about 45 minutes times 2. In that first day, but going forward, we've now convinced our IRB that we can now do this safely. So what we're going to do is we're going to give the tracer. We're going to wait about 2 to 3 days. We'll do a quick 45-minute scan, and then we'll wait 2 to 3 days and do a quick 45-minute scan. So it'll be like an hour each time of total participation, not including travel and God forbid parking in San Francisco. But anyways, it should be a good. It's good. We're making it much more streamlined. So it'll be 2 images, 45 minutes apiece, separated from the tracer injection, and that's it.
Great, because I thought that 8 hours was a little much. Great.
It is a little much, yeah. And we actually had feedback from our initial participants that this was too much, and we heard them, and we also increased the remuneration a little bit just because it was taking away from people's days that they didn't expect. But we also now have gotten to the point where we have enough information that we can. We don't want to do that again. Yeah, no, we learned the hard way that is not the best way to do the first MRI study. Yeah.
So can I ask what the remuneration was? What did they get?
I'm sorry?
Can I ask what they actually got? How much?
Oh, it's about 125 for the first day, and then 100 for each subsequent day, approximately. That was, but then now there's a bonus for if you complete all sections. I don't know. It's around the same that they do for the cancer trials. So it's about 125 for the initial scan, and then 100 for each additional. Yeah. It's not. It's a long day. It was, but we're over with that phase of the study now. It's no more.
Other questions?
Tim, may I ask 3?
Yeah.
I'm wondering, so I don't think dose related is really what I'm asking. I'm wondering how quantitative, of course I'm interested in the VRC01.
Yeah. Oh, sure.
Can you separate out Is it dose related with respect to how much viremia that people have? So a person who's at, 80K versus 20K.
Yeah, so we're doing the quantitation now, Rita, so we'll have some information for you guys soon about that. I think the challenge with the VRC antibodies, which they've also found with monkeys, is that the binding can be widely different. Even if it, quote, binds, the binding affinity can be different for each individual. So what we're also doing is we're working with Chris Petropoulos at Monogram, and what they're going to do is their affinity testing of the antibody to each of our individual participants. We've already done a genomic screen to make sure that they don't have mutations, but that doesn't mean that they have different levels of binding. So we're going to put that variable, and I think that will be very helpful. But obviously, we're going to do that on all the individuals that we image, so we have that variable as well. That's going to be critical. So I think that's a really good question. What I think we are seeing, though, is that we're definitely seeing more signal in higher viremic individuals. That I can tell you off the bat. Is it directly linear? No, but we definitely see in folks that are, say, 70, 200,000 copies, we're seeing a lot more signal in various area than we are lower, but we're still able to see signal in people with as low as 2,000. And I think that's cool.
Yeah, it's good news that it doesn't need to be linear, right? It could be logarithmic, it could be anything.
Oh yeah. I wouldn't expect, if it is linear, I think something would be wrong. Or it would be too good to be. I was like, no, that's too good.
All right,
so yeah, I'm not worried about that. But yeah, we definitely see kind of a general trend that the people with higher viremia have definitely more signal, if that makes sense. I will quantify it.
Two more quick questions. I don't want to hog all the time.
Sure.
So, of course, there's the theory about ongoing at least production, if not replication. I assume you want to use these data to support the hypothesis that there's ongoing at least production in tissue.
Oh, sure. Yeah. So I mean, if we're seeing signal that's different from uninfected controls, if we actually are seeing a difference, it suggests that, I mean, it could be that there's increased inflammation in the area, so there's more non-specific tracer uptake. That could be, I mean, certainly there's biases involved, there's some limitations, but it's certainly, I mean, would certainly be provocative. If we're seeing bone marrow glowing in people with suppressive ART, that would give us a little, because not many people look in bone marrow, right? So maybe we're not.
Collins did, and nobody believed it. But I have one more question.
Right. Well, yeah, Kathleen, right? She was like, look, stem cells, but maybe they're not stem cells. Maybe they're trafficking T cells that are going through there. So.
Right.
But either way, if there's something there.
I have a last question that's the hardest, actually.
Sure.
How many cells would you expect that you would need, and/or how many virions would they have to produce for you to see the VRC01 signal? Or how would you
yeah. Excellent question. I don't know right now. I don't know. We are, the problem is it's almost, so I can just say it's almost impossible to do that kind of correlation with modality. There's a reason they didn't do it for the monkeys or didn't report it in the Nature article when Santangelo's group did this. There's a reason that it's never reported in the cancer literature. The problem is that you can do PET tracer uptake. You can do radioactivity monitoring in tissue dishes, for example. You can do it in small animals and things like that. And so we've talked about, for example, using our labeled zirconium, slapping it on some cells, and then seeing kind of how much radioactivity each cell gives, and then trying to make a correlation. So we have actually been kind of planning some of those experiments. But I think the problem is whenever you put someone into a person, there's a huge number of variables that now start to come up, including the distribution, the metabolism. It has to do with, again, the binding, the specificity or the avidity of the antibody to the particular envelopes that are there. It has to do with the amount of fibrosis in those various tissues. And that's why we're not seeing these linear relationships in many of these studies. But so it's actually very difficult, and even an estimate could be very wrong. So that's why you often not see that type of quantitation in these types of PET studies. So it's not commonly reported because it's very difficult to do. That said, we are certainly thinking about ways to do it, because I think it would still be very important. And I agree with you. I think we need to figure out how to do that. So we are kind of playing around with some ideas. But the other problem is that it's a higher dose of radioactivity than most labs are capable of handling. So, we have this great radioactive radiochemistry lab, but it's not a BSL 2-star lab. But I have a great BL 2-star lab, but I can't do this amount of radiation in it. So, they're just technical difficulties. There are very few centers that, no one's really imaged this stuff before. So no one's really thought about, oh, if this is an infected mouse and we have to bring it into a PET MR machine that's open to all the other mice, can we actually do that? and it turns out you can't. So we've been actually thwarted a couple of times. We've been trying to work through our system here, but I think we've been, you know, I think the main issue is that it's not going to be totally quantitative, but I think that's okay. I think the fact is that we're going to see patterns and we're going to see tissues and compartments. And that's what's going to be interesting to us more so than, oh, this is 10 particles versus a million particles. I think what it's going to do is it's going to direct us to that tissue and to say, okay, now let's go to that area. And what does it show when we directly look for virus? And that where I think is it could be, at least in the short term, very powerful.
Yeah, I suspected your answer would be that you couldn't answer it, but you know, it seems I think the whole field would love to know. I'm not suggesting that your thing is tell the answer, but how many virions do you need for the immune system to see it? Yep. That would be super valuable to know.
Absolutely, not just virions, but how many surface expressed proteins as well. Right. So it's a little bit more complex than just viral particle. But it's like, are there cells that have some but not a lot of protein? Are there some cells making a lot but some with none?
Which protein?
Yeah, which protein? Yeah, right. You know, what's cool to me is that even in the lymph nodes that we've imaged, they're heterogeneous. We see areas of lymph node that light up and areas of the lymph node that are stone cold.
Why do you think that is?
What's that?
Why do you think that is?
I think it's because that there are areas that we already know just from excisional studies that there are areas like B cell follicles that are much more enriched. There are areas of lymph nodes that are fibrosed and have very little lymphocytes in them, for example. And I think what we're seeing is the natural heterogeneity of the distribution of the CD4s in the lymph nodes and potentially other cell types too. God forbid I bring up the whole macrophage myeloid thing. But with bone marrow, we may have to go there. So I think that it basically tells us that it's not just like background signal that we're seeing, that this is actually something that's going along with the heterogeneity that we expect in the tissue. The bone marrow, for example, is much more homogeneous, which would be expected given the distribution of cells, and there's very little kind of substructures or microstructures in the bone marrow. It's more homogeneous in its makeup. But we're seeing the signal in the bone marrow that's metabolically active, which is making B cells and making blood cells. That's where we're seeing that's where we're seeing the signal. So we're seeing it in areas where we would expect, if there was HIV persistence, that's where we're seeing it. In fact, and to get back to some of Rowena's question, in our one participant, actually, so one of our female participants has viral load of over 100,000, very high viral load, and her bones lit up on the MR PET machine, almost like it looked like we could see her skeleton on the PET, which was just highly unusual, but it's suggesting that there's a lot of GP120 being expressed all throughout her marrow. We don't see it in the osseous or the bony structure. We see it in the marrow, the liquid part of the bone. So that to me kind of sealed the deal for the bone marrow. That I'm already thinking about how we could get Josh to do RNA-scope and hybridization studies and look at T cells and macrophages and things like that. So we're already kind of moving on to that next stage, just theoretically until we have the infrastructure to do it. But essentially, I think that's if that makes sense. So there's definitely kind of this rough correlation between viral levels.
Amazing. Anybody else? Well, I'll tell you, that was a mouthful. I've been waiting to see this for a while. I missed it every time you did it on one of the other calls, and it's really pretty amazing.
Yeah. And we actually, for VRC, we've done 8 individuals so far. So we have 4 viremics and 4 uninfected controls. We're actually doing the formal analysis now this week, so we should be done in the next couple of days. We're very excited for that. I'm really hoping, I know what we're going to see, but it'll be nice to show it quantitatively. Again, I'm not being biased because we're having someone else doing that analysis. But yeah, no, it's been very, we're really excited about this. And if anybody wants to participate or knows people that want to participate, send them along. Well, there's a wait list already, but we can, people who, we have some wiggle room. So,
well,
yeah.
Thank you so much. I mean, we wish you luck. Let us know at DARE and Emperor if there's anything we can do for you as far as the cabs are concerned. And,
sure.
Hooray. Yeah. Anybody else before we wrap it up? It's like 7 minutes after 6. Okay, great, Tim. Thanks so much. We really appreciate your time.
Okay, no problem.
All right. Yeah. Thanks, everybody, for participating. Great call.
Yep. Okay.
Bye.
Thanks, everyone. Yep. All right. Bye.